Hi everybody, and welcome to cell structure. So we're going to go through, and I'm actually going to record this lecture so you can listen to it. So I send you the Khan Academy video. Please take a look at that because that pretty much goes through what I'm going to go through, but there might be more clear explanations. So for cell structure, so far we have done the debate. You guys have the video, and then I will also walk you again through the protein transport like we did on the board. So the first thing, of course, is cells. They were discovered in 1665 by Robert Hooke. The reason that cells are called cells is because he was actually observing cork, a piece of cork under the microscope. And when he saw the cork under the microscope, he basically saw what looked like tiny rooms. So he thought about monks and their little cells praying, and that's why cells are called cells. So now you know. Later on, there were other studies conducted by Schleiden and Schwann. You don't have to know the names, you don't have to know the dates, but basically all of these people contributed to the cell theory. So Schleiden and Schwann's cell theory is the handout that I gave you that we will be going over later in the week. So what does the cell theory actually say? The cell theory states that all organisms are composed of cells. Cells are the smallest living thing, so even if something is single-celled, as long as it has that cell, it's alive. And cells arise only from pre-existing cells. So basically this idea of spontaneous generation is no longer what is believed or agreed upon in science. So what you want to do with the handout that I gave you, please go through that, take a look at the Pasteur experiment, because that will help you understand what we mean by spontaneous generation and how the cell theory basically shows support for all of these through Pasteur's experiment. And the other thing to know is that all cells today represent a continuous line of descent from the first living cells. So we don't know exactly what the first organism was or the first living cell, but we know that every single thing that exists because of cell division, so mitosis or meiosis, which we're going to be covering soon. But the important thing is spontaneous generation is not correct. So another very important concept in biology is surface area. And as you may have seen in your textbook, if you read the textbook, cell size is limited. So what that means is your cells can only grow up to be a certain size. If you remember the microscope lab that we did, your cells were about 70 to 100 micrometers. And that's pretty small, but that is the best size that these cells can grow to. If they grow any bigger, it's going to be very, very difficult to move substances in or out of the cell. And the rate of diffusion, basically how fast things go in or how fast things leave, the rate is definitely affected by the surface area, meaning how large the cell is and how much of it is in contact with the outside. Temperature, concentration of gradient, and distance. For right now, just basically focus on this. Surface area, very important. So the surface area to volume ratio, all organisms of many small cells oh, wow, all organisms are made up of many small cells, but basically the advantage over large cells is that things can move in and out pretty easily. So this is what we were saying before. And some of the differences are that there are really long neurons in your body, and the way that they get along with being large is that they're actually very, very long and very, very skinny. So basically that maximizes the available surface area, and these organisms and these cells are able to be pretty long within our body. So in terms of biological classification, next we're moving into which organelles are found in which groups. So this is one of the things we went through on the board when we did protein trafficking. So bacteria and archaea are single-celled prokaryotic organisms. And then eukarya is a eukaryotic organism, and basically we are eukaryotes. So our animals, fungi, certain, all plants, and we are more complex. So that means that we also have more organelles. So let's get through and see which organelles are found in which organisms. So some of the basic similarities between prokaryotic, meaning bacterial or archaeal cells, and us, or eukaryotes like yeast, is that there's definitely a region, whether it's a nucleoid region or a nucleus, where DNA is located. So DNA has to be present, and there has to be some sort of a central region where it is stably there. All organisms also have the cytoplasm, and that's your basically liquid within the cell. So between the nuclear membrane and between the cell wall or cell membrane, or in prokaryotes where we don't have a nuclear membrane, it pretty much surrounds everything, and then there's a cover for the cell with the cell wall. Another important structure that's found in everything is ribosomes. And as the ribosome group told us, and as I mentioned to you guys, and as you told me, the function of ribosomes is to synthesize proteins. So what happens is ribosomes basically grab onto amino acids, which are brought to the ribosome, and the RNA within the ribosome, it basically synthesizes these peptide bonds between two amino acids. We'll go through all the details at the end of the semester, but just for right now, remember that these are the basic structures. And then lastly, there is the plasma membrane, which is made up of the phospholipid bilayer. So if you think back to your macromolecules, so far we have DNA, which is macromolecule of nucleic acid. We have the phospholipid bilayer, which is also a part of the lipid macromolecule group. And then we also have ribosomes, which are part of the protein group. So we have 3 out of 4 macromolecules just in these basic structures. So let's begin with prokaryotic cells. They are the most simple organisms. As I mentioned, they lack a membrane-bound nucleus, which means they have a circular DNA, and that DNA is found within the cytoplasm around the middle, but not exactly like it is in the eukaryotic cell. There is a cell wall in certain prokaryotic cells, not all, and they definitely contain ribosomes. Now, mind you, these ribosomes are free-floating. They are not on the rough ER because prokaryotic cells don't have the rough ER. So everything that happens for transcription, translation, building, and shuttling of proteins happens right in front of everyone else. Okay, so let's move on. So basically, prokaryotic cells, like I said, pretty much everything happens in the cytoplasm because there's nowhere else to go. As I also mentioned, there are 2 domains of prokaryotes. There's archaea and there are bacteria. And this is an example of a bacterium. So we have our pilus, we have flagellum, there are also cell wall components, plasma membrane, there's a nucleoid region, you can see that there's DNA. There are ribosomes, which are these dots, and then there's the cytoplasm, which is everything else. So they're pretty simple. Bacteria, just like everything else, has an outer covering. Some bacteria have cell walls, and they are composed of peptidoglycan. And there are different types of cell wall because there's some that go with plants and some that go with fungi, and depending on which organism you're studying will depend on what's found in that cell wall. To basically explain what the cell wall does is its job is to protect the cell, but it's also to maintain the shape of the cell and then to prevent excessive intake or loss of water. And that's very important because you don't want your cell to burst if it takes in too much water. And based on bacterial susceptibility, based on bacterial cell walls, that will determine how well these organisms respond to antibiotics, because a lot of antibiotics basically punch holes in the cell walls or cell membranes, and they allow basically these gaps to form and kill the microorganism. And then one important thing to note is that archaea lack peptidoglycan, so they're different than bacteria. The next component is the flagella, It is present in some prokaryotic cells, but not all. There could be one flagella or there could be multiple flagellas. It is used to actually move the organism, so it's used for locomotion, so the microorganism can swim one way or another. It spins in a circle, so it basically goes round and round and round and round until the bacterium gets to where it has to go. So if you can see here, this is basically the flagellum, and it can spin one way or another depending on which organism it is present in. Okay, so now we're moving on to eukaryotic cells. The biggest thing with eukaryotic cells is that they possess membrane-bound organelles and membrane-bound nucleus, and they have membrane systems. So there will be nuclear membrane, there will be membrane systems such as the Golgi, the rough ER, the smooth ER, lysosomes, and things like that. And because of all those extra structures, obviously eukaryotic cells are more complex than prokaryotic cells, which is okay. And as I mentioned, since everything is pretty much in its little compartment, that is the hallmark. That is the prime example of what will be found in a eukaryotic cell. And basically some of these systems are called endomembrane systems, so that would be a good way to remember. Because they're true organelles, they're endomembrane systems, and then there are cytoskeletal components, and we'll go through those as well. And also, speaking of cytoskeletal components, eukaryotic cells will have those, and their job is basically to expand and support the cell so that it's able to do the functions that it needs to do. Okay, so this is an example of an animal cell. I think I've showed this to you before. So the nuclear envelope is this area right here, and then you have the Golgi right here. I'm sorry, and then you have the rough ER right here. You have the smooth ER right here. You have the Golgi right here. We have mitochondria, and then we have the membrane system right around. And then you also have the plant cell. The plant cell also has mitochondria, which is good. Nuclear envelope, rough ER, smooth ER, Golgi's right over here. Has chloroplasts, large vacuole. You can't see the cell membrane, but it's technically right here. And then this is the cell wall, like it is found in every plant group. Okay, so let's begin. So for the nucleus, it's basically where the nuclear envelope protects the genetic information. So you have the nuclear region, which is pretty much a hole that is surrounded by the nuclear envelope, and right in there you will have the nucleolus. So nucleolus is the region where ribosomal RNA is found. This RNA is going to bind with proteins in the cytoplasm and help to make peptide bonds between amino acids. In terms of the DNA, most eukaryotic cells possess a single nucleus, and that is found in that nuclear region, and that is the genetic information that is stored. The nuclear envelope is made up of two phospholipid bilayers, which is pretty much what the cell membrane is made up of, but there are holes in the surface called nuclear pores, which basically control how fast and how much of something goes in or out. And then in eukaryotic organisms, the DNA is divided into multiple linear chromosomes. So like I mentioned, in prokaryotes, it is one circular chromosome. In eukaryotes, not only is there more than one, but they are basically divided into these little regions where some chromosomes have some genes and other chromosomes have other genes. A couple of words that we want to know are chromosomes, chromatin, and chromatids, and we'll get to that in just a minute. So what you can see is here's our nucleolus, here's our region where the chromosomes would be found, here is our nuclear envelope, and these little holes are the nuclear pores through which RNA can leave and go out into the cytoplasm.